Welcome to QbD in Formulation Development, presented by American Pharmaceutical Review and sponsored by ACG. My name is Mike Auerbach. I'm the Editor-in-Chief of American Pharmaceutical Review, and I'll be the moderator during today's event. In this webinar, you will learn how to build meaningful product quality specifications that are based on clinical performance, enhance your understanding and control of product and process design to increase process capability and reduce product variability and defects, and use this learning to help with regulatory approval and post-approval change management. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note, the right side of your screen also features an overview of this webinar and more information regarding our speakers. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speaker. Dr. Parizal Elchadana is the Principal Technical Consultant at SciTech Center, ACG's R&D affiliate. She is a pharmaceutical industry veteran with over 38 years of experience in R&D and setting up and running EOUs and manufacturing plants compliant with global regulatory requirements. She has managed products related to QbD and product development with successful regulatory approvals and subsequent commercialization. She has a strong technical background and IP skill set with a focus on pharma generics and innovative new product development. She has been closely associated with academia and has quite a few patents and publications to her credit. Parizad, welcome to the webinar.
Thanks, Mike. Hi, everyone, and a very good morning, good afternoon, good evening to all those who are attending this webinar. This topic is one of great interest, and it's so vast that it is difficult to do justice within the time frame that has been provided to me. Still, I have put in a lot of efforts to explain the concepts and at the same time support them with case studies. I hope at the end I will accomplish the task that I had set out for to deliver this particular webinar. So I'll be talking to you on a topic which is very close to my heart, that is QbD in product development. We all know that regulatory agencies emphasize on quality by design, and this began with the recognition by everyone of the fact that increased testing does not improve product quality, and it needs to be built into the product. To do this, it requires understanding of how formulation and manufacturing process variables influence product quality. I will be covering with you today the QbD approach, the why, the how, the quality risk management and design of experiments in QbD, QbD in formulation design, process design, and applications in clinical trials, which will be followed by a slide on conclusions. So as you can see from the format which is there on the right-hand side, QbD is not just related to formulation development, but it is also applicable to drug substance development, method development, manufacturing, bioequivalence, and finally, even the clinical trials. So let's go to the why of QbD approach. We all know that the regulatory framework is always evolving, and our friends at regulatory agencies are looking at us with magnifying glasses. There are various stages in product life cycle, from control from input out to the output, which would be the end of product life cycle. Earlier, we used to call it product development. Friends, now it has changed from development to product design. Process development has changed to process design. Along with that, if you are planning to use any device for product administration, the device design has also been brought in, followed by scale-up, tech transfers, commercial manufacture, and product lifecycle management. Now, pharmaceutical product development under QbD is covered by ICH Q8R2. It is also accompanied by what we call as PAT, Process Analytical Technology, and the various guidances are provided for that. followed by ICHQ9, which is covering quality risk management. We'll see how that applicability is so important for quality by design. And then ICHQ10, which is pharmaceutical quality systems. It doesn't end at that. We also have Q11, development and manufacturing of drug substance. As I showed in my previous slide, it's applicable even to the drug substance or the active pharmaceutical ingredient, the API, what we call it. ICHQ12, which is the complete product lifecycle management, including its applicability to stability testing. And then comes the continuous manufacturing, which is the buzzword as of today, followed by ICHQ14, where even the analytical method development and validation has been roped in. So this is the complete evolved regulatory framework which we are looking at. So what are the goals of the QbD program? Of course, the main goal is to achieve a meaningful product quality specification, which is not a specification which is developed through your product development process, but which is a specification that is based on the clinical performance requirement. This is very important. We have to keep the end product performance in mind as and when we are trying to build in the performance. But the main issue that we will be covering over here through QbD is the process capability and the reduction in product variability. Please keep this word in mind, variability. It's very, very important and it will be coming up time and again during our discussions. It also helps us to enhance the root cause analysis of any issues that may be coming up during your process or your product development, because you will have all the data available to you, which will be easy for you to find out what was the root cause of any issues that may crop up, and even handling post-approval changes which you may be required to submit to the regulatory agencies. It helps with you with the CAPA management, not just the corrective and preventive actions implementation, but currently the regulations require that the actions that you put in place should be sustainable and should be effective. So I talked of variability, and this is something which Deming has identified long time back. The problem that really we are looking at is the variability because of uncontrolled properties in the starting materials or the raw materials that we are looking at. This affects the finished product quality. Why? Because the manufacturing process is a locked process, it's an approved process and cannot be subject to any change. So any change in the raw materials is impacting the quality of the finished product. And in order to do that, what can we do to reduce the variability? We need to have a complete shift in paradigm to look at working towards right the first time and move from just compliance to real-time quality assurance. And this can only be obtained when we have an enhanced product and process understanding that will allow us to design effective and efficient manufacturing processes, and that can be achieved only through quality by design. So in order to do this particular process, perform the particular activities, what do we require? We require a complete knowledge about the product, the desired product performance, the product design that you are looking at, the process design that you are planning to put in place, and finally the process performance with respect to the capability indexes, the robustness of the process, etc. Now, the activities about the product can be achieved through control strategy, product specifications, product quality attributes, whereas the process activities can be covered through process parameters and various process controls. The whole thing is then encompassed under continual improvement, which is one of the requirements of QbD. And this you will see, friends, over here as put up in red, that QbD also focuses on continual improvement. I will not touch upon the other points because you will find them under ICH Q8 R2 regulations, and they're very simple to follow. So it is often emphasized that quality of pharmaceuticals products should be built in by design rather than by testing alone. So you saw those 4 points in the previous slide. How do we have each and every parameter covered with respect to QbD? So if we consider predefined objectives, it is defined through quality target product profile QTPP, and we need to identify the critical quality attributes that you need to build into the product. For the process understanding, we need to identify the critical material attributes and the critical process parameters. These are the terms which we will be coming up time and again during our discussion. The process control, the terminology itself is self-explanatory. You need to establish a proper control strategy. We will be touching upon the control strategy also a bit later. It should be based on sound science, and in order to make it sound, you need to have some literature input, you need to have prior knowledge, prior experience about the product that you are planning to develop, on what ultimately the aim is going to be of that final finished formulation, what are you expecting out of it, what is going to be the clinical benefit to the patient. And that's very, very important in order to build in the QTTP, the CQA, CMA, and CPP. And finally, quality risk management, because most of the decisions that you will take during your QBD program design would be based on risk assessment. Coming to the next slide, how a successful QBD program would look like. You have the formulation, and prior to the formulation, you have the materials which would be input materials for the formulation. We need to identify critical material attributes which will be going into the formulation. Say, for example, if it's going to be a formulation which is a soluble tablet, you will need a solubilizer as an excipient, and that material will be the most critical material, and its solubilizing capability will be the most critical material attribute going into the formulation. You will have to develop input material characteristics and specifications for those to meet the formulation requirements. I give you one example over here. There will be lots of other excipients going in, which will have to have defined CMAs. For the formulation, we'll have to identify the critical quality attributes that you are looking for. Say again, I mentioned about a soluble tablet, a mouth dissolved tablet. The critical quality attribute would be dissolution. It has to disintegrate and dissolve within 3 minutes flat. And that's the key quality attribute that you'll have to build into the formulation. You'll have to establish functional relationships between the CQA of the finished product and the CMA of the materials. Product characteristics should be scientifically designed to meet the patient needs, and you will have to utilize design of experiment concepts to arrive at the final formulation. Coming to the process, which is once again critical to the particular QBD that you are trying to use, you have to identify the critical process parameters that you will have to study, monitor, and set. As your QBD parameters, you will have to develop a PAT guidance. What are the PAT parameters that you will be studying, how you will be studying those. We'll take up those also a bit later. You will need some operational excellence through either lean synergies or Six Sigma, and create a culture of continual improvement. This is something which is a given. Even under quality management systems, continual improvement is something which is a requirement. Even regulators, when they come to audit you, they audit your facility, they are always looking for what improvement have you brought about since our prior audit. Finally, coming to the equipment, you can't miss them out. They are most important for your product and process. All equipments used should be validated and should be in a state of validation at the time of use. Otherwise, your whole experimental work or your complete process or even your batch manufactured, you may be asked to reject. So this is important, very, very important for our complete product development and process development. So your product has to be a robust product with emphasis on building quality into the product with QTTP. Your clinical trials should be robust. Your analytical methods should also be developed through QBD. And finally, even the personnel who are handling this should be trained on all QBD aspects and trained on all the equipments, the instruments that they're using. If any data that is found has been generated by a non-trained personnel, that data is null and void and it's not acceptable to the regulatory agencies. So what are the elements of QBD? Once again, it's a bit of a repeat. It's a slide which I have taken from one of the presentations of US FDA. Define desired product performance upfront, identify product CQAs, then design formulation and process to meet the product CQAs. This is a part of your product and process design and development. Then comes understanding the impact of material attributes and process parameters on product CQAs, identify the controls and variabilities that may be there, and continually monitor and update the process to assure consistent quality. And this gets encompassed under risk assessment and risk control. This is a very, very important strategy, and this has to be proven through your documentation in order to have a solid design of experiment and QBD documentation that you will put forward to the regulatory agencies for approval. So now that all what I have covered was theory. Let's look at how actually you will do it, because I think although many people talk about QBD, how actually the experimentation and all is carried out, many people are not aware. First point, quality target product profile has to be identified. What is the final specs that you are going to be putting into it? And those specs should be relatable to the final clinical endpoint that you are planning to achieve. Second is determine critical quality attributes of the materials which are going in, do a risk assessment and find out what are the critical process parameters and the critical quality attributes, do a design space, develop that, have a control strategy in place. And finally, once again, over here, we are coming to a point of continual improvement. So these are the various steps or the stages that you have for the QBD approach. Here we are coming to a certain definitions of quality target product profile. There is a definition provided in ICH Q8, but FDA has further extended the definition, and they have said that for ANDAs, that is additional new drug applications, which are the generics, the target should be defined early in development based on the properties of the drug substance and characterization of RLD, that is the reference product which you are considering for comparing with the with your own product on bioequivalence. So this is something which is very important, and their requirement is that if it's an ANDA which you are submitting, the QTTP should be based on the RLD, right? So finally, what we say that components of QTTP are related to safety, efficacy, identity, purity, potency. They could be critical and non-critical components, and they could be fixed and variable components. So in the following slides, we will see what are critical, non-critical, what are fixed and variable components for your QTTP. So coming to the attributes, let's see the first one, appearance, size, core embossing. This is something which can be suggested by marketing, how they want to see the product, how the appearance should be. Is it critical? Yes, it is critical, because if it's not being achieved and it's not acceptable to the patient, you may get back complaints from the market, and there could be an issue which is then indirectly pointing out to the product quality. Friability, we all know it's a very critical element. If friable tablets not acceptable. Identification, positive for the drug substance, whether it's critical or not, let's see, it needs to be identified. Assay, yes, critical. Content uniformity is critical. Degradation product, once again, there is a question mark whether it's critical or not. If it is a controlled degradation and it's not present in the original API itself, it need not be a critical element, which you will have to study. Residual solvent, once again, falls in the same bracket. Drug release, yes, very, very critical. Whether it's IR or a modified release, it's critical. drug release from half a tablet. I'm sure you all are aware that if a tablet has 4 lines, there is a requirement that you break the tablet into 2 parts and each half needs to be studied for dissolution. So drug release from half also is a critical parameter. Alcohol-induced dose dumping for extended release, yes, critical. Microbial limits, once again, there is a question mark. But what we see is that these parameters where we have put question mark, if you see there is any impact on the formulation or the process, they will become critical parameters. If you see no impact, then they will remain as non-critical. So critical quality attributes, once again, as defined by ICH Q8 is given over here, but from the point of view of FDA, the FDA Office of Generic Drugs further defines CQAs as drug products, those that have the highest potential to be altered by formulation or process variables. So if at all any attribute you see which during the formulation or processing can get changed, those are the attributes which are CQAs, and they have to be studied in depth, in detail, and strictly controlled. So they are a subset of QTPP. They are monitored throughout the drug product development. They should remain within safe and effective levels, and quality risk management should be used to prioritize the list of CQAs. So let us see how CQAs come about. You have the QTTP components where you can list everything that you can think of, but CQAs, as we saw in the previous slide, would be those which can get affected during the process and in the final finished product. Assay, yes, it's a CQA because it deals with the efficacy of the product. Impurities will be dealing with the safety of the product. Content uniformity, once again, deals with efficacy. Dissolution deals with efficacy. We all know about it. If it doesn't dissolve, it cannot produce the required effect. So this is how you can find out what are the CQAs from the complete QTPP components that we have listed out. So the QTPP components, they may or may not be a part of your final specifications, but the CQAs will definitely be a part of specification. And we all know that we set limits for these. Only if your final results are within those limits, your product is good for being released to the market. So coming to the critical and non-critical material attributes, when I say material, we are discussing here the API or the drug substance. So for this, once again, the appearance, particle shape and size, of course, it's critical because it can impact your formulation, your dissolution, release, etc. Salt form polymorphism, yes, once again, it's very important. Melting point may or may not be, depending upon whether the melting point is going to control the polymorphic form of your final finished product. Aqueous solubility as a function of pH, yes. Hygroscopicity, we are not very sure. Bulk density, it once again depends on what your product is. If it's a direct compression, it's very important. If it is a granulation, it may or may not be important. So it all depends on what's going to be your final process, whether this is a critical process parameter or not. Once again, on the chemical properties, it based on type of formulation and process, whether it will have an impact. Biological properties like partition coefficient, biopharmaceutical classification, etc. It's very important because these are the parameters which will impact the bioavailability and the absorption of the drug from the finished dosage form. Microbial limit, once again, it may or may not be important based on what formulation you are looking at. So let's see what are the critical material attributes on a risk-based analysis of the drug product and the drug substance. So drug product CQAs are in the perpendicular line where we are talking of physical attributes, assay, content uniformity, and drug release. Whereas drug substance CQAs, we have polymorphism, particle size, solubility, residual solvents, process impurity, and stability. So as you see, this is just a colored chart. It's a signaling. This is how we do a very, very preliminary risk-based analysis of what is going to impact and what is critical to your process. So if you see polymorphism definitely will be critical for your drug release, and hence it is a high risk. particle size will affect content uniformity if it's not a granulated product. If it's a granulated product, may or may not impact, but drug release, yes, but in a moderate manner. Solubility, once again, very important for drug release, and I think the rest of the parameters are quite simple for everyone to understand. So this is how we carry out assessment of the critical material attributes of both the finished product and the drug substance, how they will impact the final formulation on a risk-based analysis. Coming to excipients, we all know even excipients have a huge impact on the finished dosage form in the finished formulation. So here I have listed a few excipients which can go into your product, a tablet dosage form, whether it's a immediate release or an extended release. So if you are using lactose as one of the excipients, it may not have any impact. Starch may not have any impact, and MCC may not have any impact. But when you go to binders like starch or PVP, yes, it will impact if it is immediate release, but extended, we don't know. It all depends on the formulation. Coating material, non-functional, there will not be any impact, but functional, yes, it will have an impact. So this is the way you will first carry out your QTPP, and then go to the next slide where they will show you a complete listing of all the excipients and how they will impact your finished dosage form. So this is how we have to develop the complete understanding of the product that you are trying to formulate with respect to the API, the formulation, the excipients, and coming to the next steps of the basic product design. How will you do it? First, once again, you will identify all possible drug substance and excipients that you are planning to use. You'll use risk-based assessment and scientific knowledge to identify, the way we did it in the previous slides. Determine the levels or ranges of these attributes. This will come in through design of experiments or maybe through your experience as well. Analyze experimental data that you have, and then finally you put in place the control strategy. For critical attributes, you will have acceptable ranges, whereas for non-critical attributes, the acceptable ranges can be further investigated. So let's see a complete process from input material blending to granulation, drying, further blending for lubrication, compression, coating, and you have a coated product with you. So what are the properties of the input materials? You have to identify critical material attributes. Properties of in-process material, what happens is critical quality attributes of the previous step becomes critical material attributes for the next step of the operation. And for the process parameters for each of these processes, you will identify what are the critical process parameters that you need to put in place. As a case study, we will be doing tableting as one of the processes, and we will be having a better understanding at that stage. So identifying the critical process parameters over here on risk-based analysis once again. For the product CQAs, which are assay, content uniformity, dissolution, and impurities, and for the various stages of the process that we saw earlier. So let's remove this block and see how does it look like. So assay at pre-blending, granulation, drying is not very critical. At the lubrication stage, you are sure that you will have the final assay in place. Once you are sure of this, at compression and coating, it doesn't become critical at all. Whereas content uniformity, you have to achieve at the first stage itself. You have to maintain it during granulation. During drying, you are not going to lose the content uniformity. During lubrication, also, you may not lose content uniformity because lubricants percentages are very less. However, during compression, demixing may happen. and you may use content uniformity. But once the tablets are compressed, do you think we lose content uniformity? The answer is no. So once again, you have the green block. So this is how you do it for content uniformity. For dissolution also, I don't think I need to explain. You all will understand why granulation, lubrication, compression, and coating are very important in order to achieve dissolution. Impurities, once again, at pre-blending stage, impurities may not show up, but during granulation, they may start forming because of the various liquids if you are using for granulating as granulating medium. But during drying, there is a big risk of impurities coming in if they are temperature based. And finally, even during coating, impurities can start showing up because coating process also requires temperature for drying of the coating film. So this is, once that is set up, this is how your QbD based analysis will look like. You will have a complete system whereby which you will carry out various experiments. You will identify the factorials. Analysis will be carried out through the QbD software, whichever you are planning to use. You will get your own design space with respect to the parameters that you are studying. Say in this case, particle size, the rotational speed. You will have the experimental space and the design space defined. Any changes made within the design space defined are not considered to be reportable changes. You can go ahead with the formulation and the release of the product. Coming to the next step is quality risk management. Very, very important. ICH-Q9 identifies the quality risk management, and it states that the manufacturing and use of a drug product, including its components, necessarily entail some risk, and the evaluation of the risk to quality should be based on scientific knowledge and ultimately linked to the protection of the patient and level of effort. Formality and documentation of the quality risk management process should be commensurate with the level of risk, which is what we defined in the previous slides. The level of risks were defined with color coding. There are companies who are using actually numbers to define the risk. Like say, for example, they give 10 for highest risk, 1 for lowest risk, 5 for intermediate, and they will actually be defining the risk through numbers, where like numerical assignments will be there in order to exactly find out how the risks are being evaluated. So there are various assessment tools. ICH-Q9 provides all these tools, risk-based management through either flowchart, check sheets, etc. There is a fault tree analysis and various other hazard failure mode analysis. A lot of tools are available, and these can be adopted. In pharma practice, normally, I think the numerical one and the signaling one, what we call with color coding, is acceptable. Covering the role of risk management in development and manufacturing. For product development, you need prior knowledge, where risk assessment is carried out for excipient and drug substance design space. For process development, you need process understanding, and the risk assessment is carried out for process design space. Then you come to risk control. Risk assessment is carried out at the next stage when you are scaling up and doing tech transfer. The risk has to be controlled for the product quality, and you put control strategy in place. And finally, when you are doing commercial manufacture, you have a complete history of the previous 3 steps. You are reviewing the risks at this stage, and it is at this stage that you will use continual improvement. Now, how this continual improvement happens? Like if you have been manufacturing a product which has, as per your earlier studies, a limit of 90 to 110, but during manufacturing of 100, 200 batches, you found that the assay never goes beyond, say, between the limits of 95 to 105, you are bound by regulatory requirements to improve your limits and restrict your release limits between 95 to 105. And this is a part of an improvement process where you can always demonstrate that you have restricted your limits. And I think the regulatory agencies are quite happy when those type of steps are taken. So this is complete quality risk management. Coming to design experiments. Design of experiments is very, very important, but many a times we mix it up with quality by design. Design of experiment is an important component of QbD. but not vice versa. QBD is not equal to design of experiment. It is the mechanistic understanding of how formulation and process factors affect the product performance and quality, and it is basically on statistics. It's absolutely statistics. We as pharmacists, including myself, we don't understand statistics to that extent, and for which various statistical tools are employed by design of experiment softwares, and they give you the results directly. So there's no need to panic if you are not understanding statistics. So one needs to really understand the difference which is there between QBD and DOE. So these are the various statistical techniques that can be used for data analysis. We are all aware of process capability index and statistical process control. But when it comes to second order equations, I think even I myself get a bit jittery. So we use these various statistical packages. I think Minitab and Design Experts are the one which are most commonly used. So how do you find out what is the design space? Design space is the multidimensional combination and interaction of input variables and process parameters that have been demonstrated to provide assurance of product quality. And working within this space is not considered as a change. So this is what is very, very important and what is very useful for pharma industry. We do have variabilities, we do have variations during the process, and if we can maintain ourselves within the design space, we need not apply for regulatory changes, requirements for prior approvals before making those required changes. Now, we use the word variability in the previous slides. So variable input to controlled output is something which we are looking at. So the traditional method was we had variable inputs, but the process was fixed, so we had variable outputs, and then the drug product may or may not pass specifications. But through QBD and PAT, although we have a variable input, the process is adapted to match the variable input which is coming in to give us a consistent output, and hence you are quite sure that the final drug product will meet the specifications. So briefly, a bit about design space definitions and the regulatory flexibility, but it is important to note that the design spaces proposed by the applicant is not necessarily the full and final one. It can be changed, and the regulatory assessment can ask you to repeat it, and before they give you the final approval. We have experienced that they do come back and ask for some repeated experiments in even control the design space. They may be in different forms, like you may have a surface plot or a contour plot, or even you may have a three-dimensional plot. So in summary, normally previously we used to do OFAT, that is one factor at a time, which was a universal approach. But through design of experiments, we can do multivariate approach where you can study quite a few parameters at a given time and arrive at the final results. So overall, design of experiment is a very valuable tool to facilitate implementation of QBD. I mentioned control strategy quite a number of times. Control strategy is nothing but coming to a level, which is level 1, which is an enhanced approach where you are not testing each and everything. It is a real-time automatic control and a flexible process parameters to respond to the variabilities in the input material attributes. Whereas previously in level 3, everything was being tested, what we were talking of, testing quality into the product. Then we came to a level 2 where there was reduced testing. We'll test only the key parameters and the rest we'll assume that they are okay and leave it to the supplier. Or we see that our process is so controlled that the final product will have those parameters in control. But the level 1, which is the enhanced approach, is the final one where control strategy is applied, where you may or may not have testing being carried out for the end product, and for which here at the side you have couple of points: control of unit operations that have impact on product quality, and in process or real-time release testing in lieu of end product testing. If these two parameters are controlled, then you can reach the level 1, and these controls, they can be achieved through PAT. And what is PAT? PAT is process analytical techniques. It's a very useful tool. Instead of collecting samples, taking them to the lab and analyzing them and releasing the product for the next stage of manufacture, you have systems in place which will be testing online or inline and continuously monitoring the manufacturing, and in time give you the critical quality and performance results online to allow you to make the final go no go decision. So the focus over here is on the process and product understanding and not on advanced online monitoring of the process. So here I have a few examples of PAT. These are probes which are then put into your blender, whether in a V blender or in a granulator, and those probes will give you through NIR a real time data of whether the uniformity of the granulation or the blending is achieved or not, instead of collecting samples after half an hour, after 40 minutes to check whether the blending endpoint has been reached or not. There are other probes which are available, which are then inserted in a processor, which can measure particle size, which can measure the LOD, which can also measure the uniformity and give you results online, inline, to allow you to make go no go decisions. So they help you to demonstrate that the process is maintained within the required design space. We talked of PAT quite a number of times. This is what is PAT, is process analytical techniques. So to give you further example of a control strategy of a roller compacted product, which is a continuous manufacturing, just see at each process stage what is being done in order to control the process. At raw materials, API dispensing, specifications which are based on the product. At blending stage, you have NIR, laser diffraction to check the particle size distribution, NIR, spectroscopy for assay, and API to excipient ratio during compression. When it comes to coating, when this thing was set up, I think the method that can be used for determining coating and endpoint was not really defined. Still, we were using dissolution as a parameter for determining the endpoint coating. But now things have changed. We have got additional methods called optical coherence tomography for determining the coating endpoint as well. So this is the latest slide on process analytical technology and quality by design, where they are talking of each and every stage of manufacture, what PAT techniques can be used, and a bit more on optical coherence tomography, because this is the latest. Here you see it can literally measure and monitor the coating process by determining the thickness of the coating. So coming to pharmaceutical quality systems, it's a systems approach which helps you to consistently produce the right quality product by using QbD, PAT, and QMS. The recommendation is, along with QbD, please even install QMS in place. Why? Because it helps you for continuous improvement. It helps you with root cause analysis, impact analysis, effectiveness, and sustainability. So this is, like, once again, the complete how you will carry out the complete QbD. I will take you through a few case studies. I guess I have a few minutes remaining, where it's very interesting case study for a directly compressible formulation. It's a generic product. We need to match the innovator dissolution profile. 3 excipients were taken for consideration through experience and through study of the innovator product. MCC, cross-caramelose, and colloidal silicon dioxide. We used 2 levels, 0 to 20%, 2 to 4%, and 0 to 0.5%. And we came up with 8 experiments that need to be carried out through DOE case study. And the responses that we evaluated were hardness, friability, and percentage drug release at 5, 10, 15, 20, and 30 minutes. So let me show you. This is the results that we obtained for all the 8 trials for hardness and friability range, and for the coated tablet dissolution data this is the data required by the reference, and this data is given by our trial product. None of them are matching to what's the expected. So what do? We put it through statistical analysis, and we find that on hardness, microcrystalline cellulose concentration has significant influence, whereas the other two excipients have hardly any influence. You can see from the p-values that we have got, you can see from the graph that we have achieved. Same way for the next parameter that we studied, which is friability. We find that the change in concentration of MCC and colloidal silicon dioxide has any marginal effect, whereas cross-carbonyls, of course, will have some effect on the formulation. Percentage drug release at 5 minutes, of course, something which is cross-carbonyl a disintegrant will have effect, whereas the other two are not having any significant effect because it's a disintegrant. 5 minutes it disintegrates, the dissolution is much better, so we can see direct correlation between those. Then similarly, we have results for 10 minutes, 15 minutes, 20 minutes, 30 minutes. This all are captured through design expert, and then we come to optimization of our formulation using these three excipients, where we found MCC has a significant influence on hardness and disso. Cross-carbonyls increases disso at 5 minutes. We saw that, which may be due to the capacity to disintegrate the tablets. Further increases, not giving any further changes in the results in the disso profile. Increase in colloidal silicon dioxide marginally increases the hardness and decreases friability. And here we found that colloidal silicon dioxide, whether it's there or not there, doesn't make any difference. So it can be removed from the formulation. We arrive at the final formulation. This is the composition. We make tablets with that, and this is the disso profile that we achieve. Excellent. Absolutely superimposable. This study was submitted to FDA and we got approval in record time. Coming to QbD in process design, this was formulation. Let's look how the process design QbD can be applicable, because increased process capability and reduce product variability, right? So I'm just skipping through these slides. This is a fishbone diagram where all the critical process parameters for a granulated product are first listed down. And then, as I said earlier, I will be taking a case study of a compressed tablet at the compression stage. The input material is granules. The unit operation is compression with compression machine. We have to identify critical process parameters for that, critical material attributes for the granules, and critical quality attributes for the final finished product, which is an uncoated scored tablet. And it has embossing or a debossing on both the sides. So we have to now finalize all the parameters that we want to study, which are critical, and then identify an equation which will link those parameters. So list all possible parameters of input material, process parameters, and quality attributes, and then find out which are the most critical. Now we found particle shape is most critical, because if you would have seen in the previous slide, the granules are flowing into the compression machine from the floor above. So it's a vertical flow, and hence the flowability of the granules is very important, and particle shape we had found play a major role. The flowability is very important, and we had found that the moisture content either hampers the flow if the moisture content is high, and therefore we had put a control on the moisture content as well. So these three parameters were very important to our process of compression based on the machine and the type of process that we are planning to use. Now for the compression part, we have maximum punch load. Why? Because it's a big size tablet and there is embossing. So punch load has to be high, main compression force has to be high, and punch penetration depth has to be good in order to have a good embossing or a debossing of the tablets. Because if the embossing is not done properly, all those tablets will get rejected. Coming to the final quality attributes of the finished product, since it's a big tablet, assay may not be impacted, content uniformity may not be impacted, but impurities, yes. Impurities could have got formed during the whole process. Dissolution, very, very important, and breakability test, because as I mentioned earlier, tablets with scores have to be broken, and each broken side has to be checked for dissolution. So under DOE approach, you see CMA, critical material attributes, very important. Through risk-based analysis, critical product parameters are established and you have your design of experiment set up. And this is how the complete unit operation works. We get a three-dimensional space for this particular thing with compression force, penetration depth, and punch load. And this internal space is the design space within which one needs to work. So final words, understand and control the raw materials, API and excipients, develop and understand the fundamentals of each unit operations, crack three key critical parameters, use these parameters to characterize the project. Once again over here, look at continuous process improvement possibilities. This all make good business sense since it will reduce batch failures and simplify changes and improve inspection outcomes. One needs to implement a robot QMS. So this is a brief summary of all the steps that we discussed up till now on DOE. I'll skip that and move on to QbD in clinical trials. Clinical trials, QbD has to be performance-based quality specifications. And here I'm taking a case study with you, which has actually happened and reported by US FDA. Clinical trial protocols have to be designed to incorporate parameters that are critical to quality and through quality risk management framework. This case study is case study of a chewable tablet where there were consistent complaints coming from the patients that just in case they miss chewing of the tablet or patients who are not able to do the tablet is excreted as is and there is no efficacy seen from the tablet which has been administered. So what happens? The US FDA issued a new draft guideline covering the quality attributes when chewable tablets are being developed, and they have asked the manufacturers that these critical quality attributes such as hardness, disintegration, and dissolution, which were earlier not given any priority. Why? Because the patient is expected to chew the tablet. So why study disintegration? Why study dissolution? Once he chews, the tablet is disintegrate and the drug will dissolve. So they were not used as critical product parameters and they were just overlooked. So the agency has said that widely ranging values were not just an accepted criteria. They have to follow basic characteristics of easy to chew, palatability, of appropriate size and shape, and it should be able to disintegrate readily to maximize aspiration and facilitation of dissolution. So this disintegration is not just for disintegrating and swallowing, but if a patient cannot swallow, he should be able to aspirate it and throw it out. Thus defined the critical quality attributes, which were hardness, disintegration, and dissolution, which were missed out absolutely in the past. And these factors, they said, would also influence bioavailability and bioequivalence. And this is very, very important because of the experience that patients have faced with a particular chewable tablet formulation. So for NDAs, FDA says that the sponsor should be able to answer the following questions. Were the chewable tablets swallowed intact or after being thoroughly chewed? And if swallowed intact, does the shape and size of the tablet pose a choking or bowel obstruction risk? If water was used for swallowing, what is the volume of water that was used? And what is the experience of the subject with respect to sensory requirements, that is taste, mouth, feel, and what was the aftertaste? So if it's an NDA for a chewable tablet, all these parameters need to be studied. And if it's an ANDA, that means it's a generic product, the company needs to study sensory experience such as taste, mouth feel, ease of swallowing when conducting the bioequivalence studies. And for those companies who have not done these studies, they have asked the companies to withdraw the product, reevaluate them at their end, and if required, redevelop and take appropriate action to alleviate the risk to the public health. So a simple chewable tablet formulation based on what it is expected to do in the patient, that is have good disintegration, good dissolution, good taste, this will impact your product design. So this is what we are calling quality by design, keeping the end product in mind. Next, clinical trials, biologics and generics. This is once again my own experience where we had developed biologic, which was a generic product. We don't call them biogenerics, but we call them biobetters, and we carry out preliminary primary clinical endpoint studies where we check the efficacy of that particular product, and we also do secondary endpoint studies where we also check on the claim of the efficacy by demonstrating additional effects and supporting a causal mechanism which we are claiming for that particular product. Now, what happened? On risk-based analysis, we did not do immunogenicity studies. If we would have developed a clinical trial protocol on risk-based analysis, immunogenicity testing would have been a part of the endpoint clinical trial. We did not do that, and we had to redo the complete clinical trials once again. So the trial designs with biologic primary endpoints can determine the changes in immune parameter, which is high risk, in parallel with dose escalation to determine the maximal biological dose, or alternatively that no immunological effect occurs. This is very important, that no immunological effect should occur, and that's where immunogenicity testing is required at the time of endpoint clinical testing. So this brings me to the end of my complete presentation. So in conclusion, QbD stands as a fundamental framework in ensuring systematic development of high quality products. It has to be based on risk management and should have a very strong control strategy. You have to demonstrate continuous improvement. It's a proactive approach. And finally, embracing QbD principles is not just a regulatory necessity, but a strategic investment in achieving excellence in pharmaceutical development. So in the end, I would say that in the realm of pharma where standards align, QbD emerges with a paradigm so fine. A twist with science, a risk-based quest, quality blooms to provide a product at its best. So thank you very much for this opportunity to talk to you, and I will be pleased to take any questions if you have one.
Thanks, Parzad, for your excellent presentation. I'd like to remind everyone that you can submit any questions you have for Parzad in the Q&A box on our screen, and we'll get to as many as we can. We do have several questions in already. Parzad, our first question is, would you consider taste of an orally dispersible tablet a CQA, since this is going to affect patient compliance?
Yes, definitely. It would be a CQA, but once again, it would just be parametric because you will not be able to assign any particular rating. And taste is something which is very personalized. Like someone may like a strawberry flavor, where someone else may not like a strawberry flavor. But generally, as and when we flavor a particular formulation, it's always based upon the basic taste of the drug itself. So there are certain parameters and standards which help us to decide which flavor to put for taste evaluation. And as we put it, we do some preliminary studies and gain some intelligence into what would be the preferred taste for that product. But taste, yes, is one of the important key parameters for a particular product. It also depends upon what the product is. Whether it's a prescription product, then taste will not have much impact because the doctor prescribed, the patient takes. If it's an OTC product, then taste will be a key parameter.
Excellent, thank you. Let's move on. We have another question. While designing a robust drug product which has the intended CQAs, what are the biological properties that a product development scientist must give serious consideration to?
Biological properties that one needs to consider are a reflection of properties of dissolution and disintegration. Even things like partition coefficient for the bulk drug itself, membrane penetrability is very, very important. And on many occasions, as and when we are looking at certain different modes of administration, it's not oral, it's topical. Then you have different other parameters like skin absorption studies, which are included as essential parameters while studying the particular formulation. So it all depends on what is the dosage form that you are looking at and what's going to be the route of administration.
Mhm. Excellent. Thanks, Parzad. So moving on, we have a few more questions here. Can you elaborate a bit more on the control strategy that you presented?
That's that's a very very long response, because I just presented in a short form in the pyramid. If you remember the pyramid structure that we had, the level 3 of control strategy is total control. Everything is being tested, like all the parameters of incoming material is tested, all the parameters of process are tested, all the parameters of finished product are tested. So it is like testing of the product to have the final release. Then we come to a level 2 where it's a blend. You do reduce testing, certain non-critical parameters are not tested, whereas critical ones are tested and the products are released to the market. And then secondly, we come to the level 1, which is the best control strategy, where we are talking of using initiatives like PAT initiatives, where testing is not done of the end product. The critical material attributes are inbuilt into the material. The process goes through with all the PAT initiatives. I did show a couple of slides on the latest advances in PAT. You have those initiatives in place. You do not test any of your in-process material. The process just flows through and final finished product can be released to the market without testing. So this is the control strategy where you are assured that your process is under 100% control.
Excellent. Thanks again. All right, let's move on to our next question. Does one need to inform the regulatory agencies if there are variations within the design space?
If there are variations within the design space, one need not inform the regulatory agencies to obtain prior approval before incorporating the variations and releasing the batch. You can proceed with those variations and the batch can be released to the market, although the variations were observed. But those variations need to be noted down, and you need to inform the regulatory agencies at the time of putting in your annual submissions. I hope all of you all are aware that for each product, each strength, we are putting annual submissions to the regulatory agencies in whichever market you are marketing those products. In those annual update, you will have to mention the batch number and the product and the strength for which that particular variation occurred, and the fact that you released it to the market and why you released it to the market, because it was within the design space. So yes, you need to inform the regulatory agencies, but no, you need not wait for their approval in order to proceed with the product.
Excellent. Thank you. Our next question is, can you provide some inputs on QbD for API development and manufacture?
I'll keep it very brief. It's very similar to what we are doing for the finished dose. In API also, we have starting materials. So you have to define the critical quality attributes of the input starting materials, and you have to define the critical quality attributes of the finished product. Now, during the process, if say, for example, you are heating at a particular stage at temperature of 50 degrees in the process with continuous stirring, and you know with heat a particular impurity can get formed at that particular stage, the control of temperature is very very important. So that will be your control strategy, your CPP, right? So this is how you define the input material quality. The output material quality, when you are defining, you will put that particular impurity as a limit, which is the lowest possible that you can achieve. And if it is an identifiable limit, then the pharmacopoeias allow you to up to 0.2%. If it's not possible to identify and characterize it, the pharmacopoeia allows you a limit of 0.1%. So accordingly, those limits can be put as your final CQA in the finished product, and your CPPs have to be managed in such a way that those limits are achieved. So this is how you characterize your impurity. This is how you find out your final finished product specification. Not that you start your process and you put whatever uh, results you achieve as your final specifications. Your final specifications have to be designed based on what you have to achieve. So this is how you will work for a API or a bulk drug.
Excellent. Thanks again. Moving on, our next question is QBD now extended even to analytical method development?
Yes, I did mention on a couple of occasions during my slide deck that QBD is also extended to method development and validation. I will take a simple example of, say, developing dissolution method. So when we are developing dissolution method, you cannot just develop ad hoc any dissolution method that's giving you a good disso profile for your particular product. The method has to be biorelevant. That means whatever results that you get out of the dissolution of that method, you should be able to have an in vitro in vivo correlation with your bio study. So it has to be a biorelevant method. And some of the critical process parameters for a dissolution study would be things like volume of the dissolution media, would be things like speed of the stirrer, would be also things like the pH of the medium, the type of medium, whether you'll use acidic medium or you'll go for a buffer, or whether you will incorporate a solubilizer. Like many a times SLS is used as a solubilizer in the disobedium. So all these decisions will be based on the endpoint that you are trying to achieve and not on the type of diso profile that you are obtaining. So the dissolution method has to be developed on the basis of QBD with the endpoint in mind, which should be clinical relevance.
Excellent. Thanks again. Our next question is it possible for a product to have two specifications, one for real-time release based on online measurements and another for end-of-life testing?
Yes, definitely. You can have two specifications. The end of life testing would be the stability spec, and that would be the final spec which you should submit to the regulatory agencies. And real time testing, which you are using for online release, would be your in house specifications. Normally, we have these two specifications. The in house specifications are specifications for the company in order to take decisions for release of the products, and those specifications are always narrower than the final specifications which are submitted for the regulatory agencies, so that at the end of shelf life you are still within those specs and your product doesn't fail.
Excellent. Thanks, Parzad. Our next question is when you do a DOE of formulations, will the process be the same in trials, clinical trials?
It has to be, because you cannot have a product developed under a different method for clinical trials and then have a different method under DOE for further scale up and manufacture. If the process changes and you have some change in the final finished product characteristics, then your complete clinical trials could be a failure. So once again, it depends on what clinical trials are you aiming at. Are they phase 1 clinical trials, phase 2, phase 3? Because by the time you come to phase 2, your formulation, your process, etc. have to be in place. So if you are coming up to phase 2 clinical trials, your complete process development using DOE has to be in place, because your final finished product CQAs are based on what you are trying to achieve in clinical trials.
Excellent. Thank you. I believe we have time for one more question. Will it be possible to widen the limits for an approved product and process specification if, post-approval, such changes are found to have no significant effect on product quality?
That's a very tough one to answer. one cannot say a blanket yes or a no. It depends on the amount of data that you have generated. You will need a lot of tox data. Just having a data that the product is stable and it's meeting the specifications, etc., it's not enough. You have to have toxicity data for the wider specifications that you are trying to get approval for. So it all depends on what the product is and what is the risk behind widening the specifications. On risk-based analysis, it may be a go no go situation.
Right.
That's my understanding. I'm not sure regulators how they will think, but if I would be in a regulator's shoes, this would be my understanding.
Great. Excellent. Thank you. Well, I believe that's all of our questions and actually all the time that we have today. Any unanswered questions will be answered via email. We'd like to thank Dr. Elchidana for sharing her knowledge with us and also offer a special thank you to ACG for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleagues. Thank you for attending the webinar and enjoy the rest of your day.